Nice. The depressed AL Bio kids are back. Assalamu alaikum. This is part 2 of the AL Bio Chapter 1, Cell Structure, covering magnification. Measuring size and calculating magnification is pretty much just math you'll need to know for this chapter. We're looking at magnification first. When we talk about magnification, we're thinking about microscopes. You know when you view things with a microscope, it becomes larger, right? Yeah, so it's basically magnified. Now we know we can have different zoom levels or magnification levels in our microscope when viewing something. And yeah, that's basically what we're looking into in this part. When you want to do anything magnification, I want you to remember this formula first. M equals I over A. Reason being, it helps really understand the definition and it'll become simpler to remember. The definition here is the number of times larger the image of an object is than the real size of the object. If we try to make sense of this definition, we remember, oh yeah, we're viewing, a potato cell, for example, through a microscope at, let's say, 20 times zoom, or more appropriately, magnification. So we're technically seeing it 20 times larger than it actually is, right? This 20 times zoom. Or if we use that 20 value, let's say, into the definition, we get the image of the potato cell we're seeing is 20 times larger than the real or actual potato cell. Please tell me that explanation made sense. Then we get to the formula. I personally memorized M equals I over A, as you see here, and this is what the triangle looks like. I know some others memorized this AIM triangle. They're both the same thing, just go with whichever you like. I know AIM is easier to remember because it's like, an actual word, so you might prefer that. M stands for magnification, I is image, and A is the actual size of the object. If we substitute the meanings into M equals I over A, we get this. Magnification is the observed image over the actual size. Straightforward enough. I'll just try to speedrun how to use these triangles because I'm very positive almost all of you are familiar with it. But why not for the sake of those who don't know? If you want to use these triangles, you basically cover the letter you're trying to find. Assuming you want to find magnification, M, in the question, you cover M with your finger or use your head and see that I and A are left. Because I is over A, it means I is divided by A, I over A. Looking for A, actual size, cover it, you get I over M. Looking for I, cover it again. You see A and M beside each other, which is just multiplication. The reason both triangles are interchangeable is because A times M and M times A are the same thing. Easy. If you decide you're more of an, veteran math user, you could memorize M equals I over A, and we'll rearrange the formula. No problem, it's all the same thing. Possibilities are endless. Lord, I spent so much more time than I thought I would on this. Okay, let's put this knowledge into a past year question now. We're given an image of a plant parenchymal cell. Cool. The actual diameter of the parenchymal cell in the figure along XY here is 35 micrometers, correct? Now they want us to find the magnification. Direct question, we can quickly use what again? The formula, or the triangles, same thing. I'd suggest attempting this question while the video is paused to actually practice and see how much you actually understood from what you just learned. And because of proportion problems, the measurement of XY with the ruler is 7 centimeters. And if you don't get it right, don't kill yourself for it, especially if it's your first time too. It's just like that sometimes. Okay, so what I did here was write down the formula first. Good. If you use the triangle, you would get image over the actual object too. To find I, we should have measured XY with the ruler because that's pretty much the only way you can find the size of the image we're seeing. Now 7 centimeters to millimeters is times 10, so 70 mm. Good for you if you use the mm part of the ruler or can do the cm/mm conversion without writing it down because it'll really help you with time. And if you remember that since 1 millimeter is 1,000 micrometers, 70 mm should be times 1,000 to get the value in micrometers. So we'll get 70,000 micrometers. Now the reason we're converting to micrometers is one, again, because these cells are super tiny, we're almost going to always measure them in micrometers. And secondly, when we do the calculation, we want all the values to be in the same units. If they're both, for example, length measurements, like this. So yeah, we got our I, now we need our A, which fortunately is given to us here, 35 micrometers. So if we slap them into the equation, we get 70K over 35. both in micrometers, of course, smash that into our calculator, and voila, 2000 magnification. And oh, it's given to you here, but this times symbol right here is the, let's say, unit of magnification. So when you want to write a magnification as 60, you actually write times 60. Another quick example before we leave. The question simplified is that the magnification is times 5600 mag. Find the length of the labeled chloroplast. Just quick skim, this is a plant cell clearly, see the large backbone and chloroplast all around, with the nucleus and nucleolus here. You may pause again if you want to attempt, and the measured length of the chloroplast is 2.5 centimeters. Yeah, anyways, again, first step, use whichever formula you want. Write it down first too, even if it's magnification isn't what we're trying to find. Because number one, it'll help you not stay stagnant and help you progress into the question if you feel intimidated or confused. Two, you get marks for just writing the formula in some questions. I'm not kidding. And lastly, just in case you might mess up rearranging the formula of m equals I over a. We're looking for a actual length. Make a the subject and we get I over m. Try using the aim triangle too and cover what we're looking for, which is a, and you also get I over m. Measuring I with a ruler, we get 2.5 centimeters, and times 10 of that, we get 25 millimeters. I'm going to quickly mention this. Please don't forget what you're trying to measure. The question states the labeled chloroplast. We have many here, and I know it's hard to see because I have the images smaller here, but the labeled chloroplast is here. Don't go measuring something else. 25 mm, and to convert mm to micrometers is times 1000, so you get 25,000 micrometers. Now we need m, which is given in the question, thank God, times 5600. Now slap it into formula and we get 25k over 5600. Smash that into our calculator and you get a as 4.64 micrometers to 3 significant figures. Yeah, measuring cell size. Honestly, so cool that you can measure these tiny cells with a microscope. What's not cool though is that A-levels is going to force you to do it. I'm going to dive straight into it with an example because it's kind of complicated. You have an eyepiece reticle or EPG, this transparent scale on the eyepiece of a microscope. The eyepiece is what you put your eyes onto to see through the microscope. This right here. Since you're viewing the sample with this scaled eyepiece, you'll get this like superimposed. Oh, so when you view the sample, you can see both the EPG scale and the sample at the same time, even if the scale is above the sample, like it is. Forgetting everything else, let's look at this cell here. Sorry if it's not large enough, but the EPG has a scale from 0 to 100, and this cell's diameter here is between 40 and 60. 60 minus 40, you get 20, so it's covering 20 eyepiece divisions. Some people call it units, but whatever really, I call it divisions. Again, covering 20 eyepiece divisions. When you get the EPG divisions, you want to calibrate the EPG using a stage micrometer. Just chill, I'll explain what that means now and hopefully you'll get it. So you know how we viewed the sample and that one cell took 20 EPG, right? Yeah, thing is, the EPG has no units. I'll explain why later, but if you're able to think of the reason before I explain, I congratulate you, really. So after we get the 20 EPG value, we take out the sample of cells and place in the stage micrometer. Now thankfully this guy has units, and what we'll do with him is calibrate the EPG. When you place the stage micrometer and view it with the EPG, you'll be able to see both scales, right? Scale here and scale here. And it'll look a little something like this after adjusting the stage micrometer to the right place. From here onwards is somewhat simple math for some. This one, 0 to 100, is the EPG, of course, and this one is the stage micrometer, which comes with a scale, and is usually given to us. Like this one, it says each division is 0.01 millimeters. Just to confirm that real quick, if this is 0.01, 0.02, 3, 4, 5, 6, 7, 8, 9, and after 0.09 is 0.1. Yeah, it's legit. 20 EPG here, remember, on the stage micrometer translates to... 0.05 millimeters. And we want to change this to micrometers again, which is times 1000, and you'll get 50 micrometers, and that's your final answer. the actual diameter of the cell was 50 micrometers. Now, to answer the question of why doesn't the EPG just have scales? We wouldn't need to calibrate or calculate, etc. Just a quick FYI, this is so not part of your syllabus, so don't worry about it. Unless, I don't know, Cambridge decided to add it to the syllabus by the time this video is a few years old. But I hope you have, and if you have dealt with the microscope, you know the eyepiece is here, and you view through it. And these three lenses, called objective lenses, have different magnification levels. And you can actually switch between them to view the sample at different magnifications. Assuming the EPG has a scale, okay? One division is 1 micrometer. If we view the cell with 4 times magnification, I know the magnifications are stated in this image, but just ignore it. 4 times magnification, and we measure, let's say, this organism's diameter, okay, diameter, and we get 50 micrometers. Okay, fair. But if we use 10 times magnification, which is larger magnification or zoom, but the EPG doesn't align with that zoom, it can't. It stays the same, and when we view the organism with a larger magnification, we now suddenly see that the 50 micrometers only covers part of its body. Like, we can't tell if the body is 50 micrometers or not, and it probably isn't. It's basically not consistent, and I don't want to explain too much about it since it's not important to your syllabus, but I hope you get it. Let's just do one more example before the video ends, because this is crucial for you to understand. This is an alternate method because the calibration comes before viewing the sample. Just gonna speedrun this. If we view the stage micrometer first, we see that all 100 EPG divisions are covered by 0.25 millimeters on the stage micrometer. To get 1 EPG, we divide both sides by 100 and we get 1 EPG is 0.0025 millimeters. Now, kind of an ugly number. Let's convert it to micrometers, so multiply by 1000 again. And yeah, you get 2.5 micrometers for 1 EPG division. Just skipping the part where we measure the sample, but if you remember, the cell diameter covered 20 EPG divisions. So to get the cell size, we multiply the 2.5 micrometers as 1 EPG by 20, and we get the same answer again, 50 micrometers. Good lord. With enough questions, you'll get the hang of it, and I dare say love the questions even. I wrote these two things down on the right because they're things you don't want to forget answering these kind of questions. First is a silly mistake many students tend to make, which is the conversion to micrometers, either during calculation or as their final answer. And secondly, you shouldn't worry about which method to use. Honestly, I interchange between both based on feel because once you get what's going on, you don't even need to think of them as two separate methods. But if you want help choosing which to start with, I'd say choose which felt easier to understand. And with that, we've reached the end of the video. Thanks SlidesGo for the template, these guys for the images, and any questions, just drop them in the comments below. Want me to use more past paper examples, or just simply use more images, just drop them below. I really would appreciate the feedback and would definitely take it into consideration. Bio is the best science, no debate. Peace and assalamu alaikum.